I'll try to be as simpler as possible. I'll share my screen, Bruno.
Yes, can you share? Do I have to auto?
Mm-hmm.
Yeah,
no, I think I can. Here you go. Do you see my screen?
Yeah.
Okay. Let's see. Okay, so I'll do this. Okay, so this is what I've been talking about. I will not tell anything about our collaboration yet, Bruno, but perhaps we can do another chat on that when we will have like the final version of our results. Okay. But that will be a great, this talk will be a great introduction of all the work that I've been doing on the interlaminar astrocytes, these astrocytes that Bruno was talking about. So you can place, if you want to see my face, if you don't, never mind, you can place the Zoom window here, and I'll make sure that there's nothing in the slide, usually in the top right corner every time. So, okay, where can I, okay. 1 second. I cannot see it, my full screen. Okay, do you see now? Good. Yes. Okay. Yeah. So you know already, because you work with Bruno, you know already what a cerebral cortex is, and it's a part of the central nervous system, and it's the place where a lot of neural integration, both sensory and motor integration, happens. And it's somewhere, somebody calls it the place of higher cognitive functions. I just call it the place of complex cognitive functions in mammals. And usually mammals has 6 layers. And this is important to remember when I will show you the morphology of the astrocytes that I study. So usually the layers in the cerebral cortex are called, are numbered from the most marginal layer, which is the molecular layer, layer 1, and up to the bottom. So the deep layers are up until the layer 6. So the neural stem cells in the cerebral cortex are also called radia cells. So that is a bit confusing because in the past they were called, when nobody really knew what glia was, they were called radia because they were thought to be glia-like cells, but actually they're neural stem cells. They're just called radia, and they can give rise to all the variety of the cells of the central nervous system. So on the one side, they give rise to neurons first, usually in mammals, and then they can give rise to glial cells. And probably you're mostly familiar with neurons, but I've focused my whole career on glial cells and specifically on astrocytes. There's also other types of glial cells like oligodendrocytes, microglia, but I've been studying mostly astrocytes since the beginning of my PhD and then also postdoc. So what are these astrocytes in general? They are these star-shaped cells, and before they were thought to be only structural cells, they sent projections to both neurons and blood vessels. So people thought that they were having just a mere structural role, but now we know that they have a huge variety of roles. So they can provide, for instance, metabolic support to the other cells of the brain, and they collaborate with the blood-brain barrier. So they take up nutrients and they redistribute nutrients to other cells. They are actively involved also in the neural transmission because they can reuptake or release neurotransmitter at the synaptic cleft. And they regulate the ion balance extracellularly. And like I was telling, they also modulate the synaptic transmission and also the synapses development during brain development. So they are pretty important cells. And what I would like to stress from the very beginning is that, like for the neurons, there's not only a single type of astrocytes. So there are many different types of astrocytes. So we're still in the process of understanding in the community of astrocyte people that what are the different types of astrocytes, how much they can be heterogeneous. So there has been a lot of recent works also on genomics and transcriptomics of astrocytes. And so the situation is even more complicated than what I'm going to explain you right now. So classically and historically, they recognize 2 major types of astrocytes, which are the protoplasmic and the fibrous astrocytes. So these were the first one that were really studied. And even now, this is a pretty broad characterization that we still use in the field. So the protoplasmic astrocytes are mostly present in the gray matter. So in the layer from 1 to 5, let's say, in the cortex. And then the fibrous astrocytes are mostly present in the white matter. I'm saying mostly because the division is not that like discrete like it seems. Sometimes you can find both in the gray matter or white matter. And also you can recognize them by morphology mostly. So there is not really specific markers to recognize protoplasmic and fibrous. So you see that the protoplasmic ones, they have more processes, and the fibrous ones, they have less processes, and usually the longer processes. So these two types of astrocytes have been studied in all mammals, and they have been found in all mammals. So we know that all mammals have these types of astrocytes. While there are other types of astrocytes that can be specific to some mammalian group. First of all, the interlaminar astrocytes, they were thought to be specific to primates, and these will be the topic of the focus of my talk today. And then there are also other types of astrocytes that are called varicose projection astrocytes that have been observed in human only so far. I also have a project on varicose projection astrocytes going on right now. So we also know that that is not totally true, that they're only present in human, probably also in great apes, but that is a whole different story. So I will not talk about these other astrocytes today. I will just focus on interlaminar ones. Okay, so why are these interlaminar astrocytes so special to me? First of all, I started to study them because everyone in literature was saying that these are primate-specific cell type. So I wanted to understand, why do primates have this specific type of astrocytes? So what's special about them? What do they do and how they look like? Is it true that only primates have them? So first of all, you can recognize their specific morphology, which is very different from, like Bruno was saying, from all the other astrocytes in the cortex. So they have a cell body, which is here, and I've highlighted it with these red arrows. So the cell body is in the layer 1, very, very close to the pia surface, just the most marginal layer here. And they send down these very long processes that are called interlaminar processes, because they're able to cross different layers that are also called lamina. So that's why interlaminar astrocytes. And they can cross many different layers, and they can reach up to layer 4 in primates. So they can go pretty down in the cortex. But remember that the cell body only stays in the layer 1, very, very close to the pia surface. Nobody knows what these processes do or what these interlaminar astrocytes do, so we don't know the function yet. They have been proposing in literature that they can help the columnar organization of the primates, which is more prominent in primates in respect to other mammals. And most of the work that was done on these interlaminar astrocytes was done by Colombo group in Argentina 15 years ago, I think, before I started my postdoc. Then really nobody really spoke about these interlaminar astrocytes for a while. And now I think they are again gaining some attention. So he studied these astrocytes mostly in primates. So he did pretty extensive studies of these interlaminar astrocytes and characterization of their morphology, mostly in primates, in a lot of different primate groups: New World monkeys, Old World monkeys, great apes, etc. But you can see that they were already observed by Ramon y Cajal and Jessen a long, long time ago. So some other anatomists already looked into the cortex and saw them before. So that was really fascinating, because they have such a peculiar morphology. So how can we recognize these interlaminar astrocytes? So we don't have a specific molecular marker yet, protein, that we can label to see them, but we can stain the sections with immunofluorescence or enzymatic staining anti-GFAP. GFAP is a protein that usually is expressed also by other astrocytes. So it's not specific to these astrocytes, but it can help to recognize their morphology. So we use this anti-GFAP staining, and then we look at the morphology and the position in the cortex. So far we can only do studies in slices from real brains. So we cannot do stuff in vitro because we don't have a specific marker. So we would not be able to recognize them from the other astrocytes. So at the beginning of my postdoc, these are the very first thing that I did. Now it's more than 3 years ago. And we really wanted to know, are these astrocytes really astrocytes, or are these other type of cells that are not really astrocytes, because they have this very weird morphology. So what I did was to use different astrocyte markers to see if they express also astrocyte markers, because nobody really tested them before. So we tested, and I don't want you to remember all the details here. We tested other astrocyte markers like vimentin, APC, AS100 beta, aquaporin 4, and they do express all these other astrocyte markers. And then we tested negative control. So we tested other cell markers. So for instance, OLIG2 and SOX10 are markers for oligodendrocytes. They do not express these markers, and they do not express neuronal markers either, like neuronal Nonhuman or MAP2. They do not express it. And they do not express IBA1 marker, which is the marker for microglia. So they truly show astrocyte identity. We can keep calling them astrocytes. That was just a confirmation that they were really astrocytes. Also because the GFAP marker usually stain in the primates also stem cells. So we wanted to make sure that they're truly mature astrocytes. So these were the three main questions that we really wanted to address at the beginning of my project. First of all, we wanted to know more about their evolution. So like I was anticipating before, when did they appear during evolution? If it's true that they are present only in primates or not. And then, as I have a background in developmental neurobiology, I was really interested in their developmental origin, how they are born, what are their cell of origin, and when they are born during the development, prenatally or postnatally, for instance. And finally, what do they do? What is their function? So I'll tell you today, we were able to answer to most of the first two questions, and we're working right now on the function, actually. So I'll tell you also something more about the most recent results that we're getting on the function. So first question, very simple. How are these interlaminar astrocytes really specific to primates? So whenever you will see ILA, this is my acronym for interlaminar astrocytes. So are these astrocytes really specific to primates? And in order to understand that, what we did was to collect as many species, as many brains as we could from a lot of different orders across the mammals. So we did that in collaboration with other professors, Steve Noctor, Chet Sherwood, Paul Munger, William Hopkins, and Patrick Off. And I would like really to thank them, because this was a great start of my PhD. They helped a lot in providing us a lot of tissues and brains and sections. So we were able to harvest the tissue from 46 different species. And these species encompassed different orders. So my idea at the beginning was to test as many orders as we could, so that we could diversify our investigation and we could look at as many unrelated orders as we could. So this went pretty well in the sense that we were able to harvest 46 species. And as you can see here, we have pretty diverse orders like elephant. We have like species like elephants or Chiroptera, which are bats, carnivores, for instance, the cats or tigers, and the artiodactyls, as you know, there are, for instance, the sheep or the cetaceans that are here, then rodents, and then of course, a lot of primates that we knew already that we would have found them in the primates. So what we did was a very simple thing. We stained sections from comparable anatomical regions, anti-GFAP, which was the protein that we knew that they expressed this protein, and then we went to look in the layer 1 to see if we could find them and if we could see these long interlaminar processes. So what we found at the beginning was pretty astonishing, was that all the mammals actually have a certain type of interlaminar astrocytes. So there is always something that resembles interlaminar astrocytes in the layer 1. So some have what we called the rudimentary interlaminar astrocytes with a cell body and in very similar in shape and position in the cortex to the typical interlaminar astrocytes, but with processes that are much shorter. So they tend to go towards the layer 1 to 2 boundaries, but they never cross it. And for instance, these are the ones that you can find in the mouse and in many other orders too. And then some other orders and species, they have what we call the typical interlaminar astrocytes, so with proper interlaminar processes crossing the layer 1. But even these ones, the typical ones, are not specific to primates. So for instance, here I'm showing you a interlaminar astrocyte, a typical one from this weird guy here, very cute guy, a rock hyrax, and this order, this species is in an order which is very, very far from the primates. So the question became, what is really specific to primates then? Why have they studied so much in the primates in respect to other orders? Is there something special in primates? And we want to look for their density and morphology across evolution. So we use the very same species that we use to investigate their presence. And we measured, for instance, the linear density that we call linear density because we did that measurement on the sections, and we counted how many interlaminar astrocytes we could find aligned on a perimeter of PS surface. And first of all, we found that the primates have more of this interlaminar astrocytes. So they have a higher linear density of interlaminar astrocytes in respect to the other orders. And then we wanted really to reconstruct the morphology, because just by looking by eye, we could see that the processes were much longer in primates than in other orders. Even when in other orders, we could find interlaminar astrocytes, the proper ones with processes crossing layer 1. So we used this software, which is called Neuralucida. And basically you put the slide, you do live imaging, you do live reconstruction in real time. So you put the slide under the microscope, and then there is a software connected to the microscope that you can use to draw the outline of the silhouette of the cell that you're interested in. So you can do single cell morphology reconstruction, and then you can analyze different things, different parameters. So you can analyze the number of primary processes, so the number of total branches, so total processes, then the total length that they cover with their processes, and also Neuralucida, they give you a complexity index calculated based on all these parameters. So these are just some examples of reconstructions of these astrocytes, and you can already appreciate that the primates have the highest morphological complexity compared to all the other orders. So you see artiodactyls here, pretty long, but not as complex as the primates. The same for the carnivores here. So when we look at the numbers, we see that they have more total processes, so more branching. They have longer processes, as you can see here. And finally, they have the highest complexity, highest morphological complexity when compared to the other orders. And within primates, if we go to take a closer look to the primates, so these are the primate species that we could gather for this study. So we had prosimians and tarsi that are the most ancient primates, as you can see here, and then New World, Old World monkeys, and great apes, and including also the human. So great apes are the closest relatives to humans. So when we look at that, we saw that among primates, the great apes show the interlaminar astrocytes with more branching and longer processes. So as you can see here, these are the ancient primates, very simple, very, very similar to other non-primates order. And then New World monkeys, we see an increasing morphological complexity, which is not that much different in the Old World monkeys, but then it becomes very much complex in the great apes and human. And we didn't find any difference really between great apes and human. We also have only one species from human, so that's also different to tell. It's difficult to tell, but we couldn't find any appreciable difference in morphology. So to conclude this first part, what we found for the evolutionary part of this study is that all the mammals, even the marsupials that we had, show at least a rudimentary form of interlaminar astrocytes. And even the typical interlaminar astrocytes are not specific to primates. And so we concluded that the interlaminar astrocytes may have a very ancient origin during mammalian evolution. Then something happened in the primate lineage that allowed them to become more numerous. So we found a higher density and also more morphologically complex, is that we found that they have a higher morphological complexity in primates and within primates in great apes. Moving to the second part of my talk, we wanted really to study the development of these astrocytes. Again, we could not, it was very difficult to think about in vivo experiment, like lineage tracing experiment that you usually do in mouse to trace a lineage to understand how they develop. So what we did was to collect different sections at different developmental stages. So we were able to collect a lot of prenatal and postnatal developmental stages, and we went to look in those specific stages during development. So we did that for 3 primate species: macaque, the rhesus macaque, chimpanzee, and human. And we also wanted to compare. So remember that these primate species, of course, they have the typical form of the interlaminar astrocytes. We also wanted to compare with the rudimentary interlaminar astrocytes that we found in mouse, because you know that mouse is one of the most studied animal model in neuroscience. So we wanted to know if we could model some of the features of these interlaminar astrocytes in the rodent as well. So I will focus mostly on the macaque today for lack of time, but if you have questions on chimpanzee and human, I'm happy to share with you what we saw also in those other species. So first thing that we did was to do exactly the same thing that we did in the evolutionary part. So we measured their density and their morphology during development. So how can you recognize them during development when they have a shorter processes yet? Well, you go and look again at the cell body, which is very, very close to the PS surface, and the processes that are pointing down. So this is an example of what they look like prenatally in the macaque, very, very short processes. And then this is at 1 month after birth in macaque. So they grow these longer processes. So we were able to count, to measure their density, and we found that the density progressively increases over age, and they are already present prenatally. And this is how the morphology also increases throughout development. So this is an example, again, examples of neurolunar reconstructions in the macaque. So gestation day 150 is right before the birth. And then these are all the postnatal stages until the adult. So it seems like the morphology reaches a plateau around 3 months in macaque, but then here in chimpanzee and human, they keep growing in morphology. This is, I won't go into details of these. This is just to show you the numbers of the different parameters that we measured. So number of branching, the ends, and the total length increases over development. And of course, the complexity too increases over development. And so when we wanted to know more about the developmental origin, so when exactly do they appear in development, and which are the cells that they originate them, we, first of all, we wanted to test them for other molecular markers that are really important to the development. So we test them from some radioglia cell markers, so stem cell markers, and again, astrocyte markers. And we wanted to know how these markers change during development. So we took the very same stages of development, so both prenatally and postnatally, and then we labeled them for all these different markers. So I won't go into details of all these markers, but some of them, like for instance, this PAC6, SOX2, and nestin, they are pretty broad stem cell markers, but they also retain in some cells in the adult. And then CREAB and HOPPX are markers for something that we call inner radioglia cells and outer radioglia cells, that are two layers of stem cells that are more visible in the primates, but are present also in other mammals. So what we found was that these interlaminar astrocytes in macaque, they express both stem cell and differentiated astrocyte markers. And as we tested all these markers for radioglia cells, PAC6, SOX2, Nestin, CREAB, HOPX, I'm just repeating the ones that I showed you in the previous slide. They do express all of them from the very beginning, and some of them, they retain the expression also in the adult. We concluded that they might emerge from these radioglia cells, so like most of the other astrocytes do. And then finally, they also express S1 and beta. We just confirmed that because we knew that in the adult also they express S1 and beta. We checked if they do express S1 and beta in prenatally, they start to express it. 30% of them express it prenatally, and then all of them express it postnatally. So this means that they are differentiating postnatally mostly. So when we wondered and we asked ourselves, do these interlaminar astrocytes develop prenatally or postnatally? We knew that we observed towards the end of the pregnancy, some of them. So we really wanted to make sure that they appear prenatally. We did an anti-GFAP staining, but it was very difficult to distinguish them from the radioglia cells that are close to the ventricle, because they do express, like I said before, also this marker. So the processes of the prenatal interlaminar astrocytes could not be distinguished from the processes of the radioglia cell fibers. And so this is why we use the anti-S1 and beta marker. So we know that some of them express it prenatally. So we just needed to look at when they would reach layer 1. So this is a macaque and gestation day 68. You see they're not really attached to the PS surface, so you have to imagine that the PS surface is on the top. And then you see that the somas are already present in layer 1, gestation day 84. They grow, they attach to the PS surface around 100, gestation day 100, and they start to grow processes in between 100 and 123. And this is when exactly they start growing longer processes. And we did the same for the human. And in human, the somas are already present gestation week 19, and they grow processes around gestation day 31. So next question that we asked was, do they proliferate across development? Usually other astrocytes, they're not like neurons. So neurons usually proliferate before they migrate. Astrocytes first migrate and then they proliferate locally when they reach their final destination. So we checked if these astrocytes too, they do the same. And actually they do, because we found that they express a proliferative marker, which is Ki67, gestation day 80, 109, but then we couldn't find any proliferative cell interlaminar astrocytes after gestation day 123, which is exactly when they start branching down the processes. So that makes sense. They arrive there, they proliferate for a while, and then they start branching down processes, they start proliferating. So this is our model of interlaminar astrocytes development. We still don't know if there is, so they most likely derive from the typical radioglia cells, which are the stem cells close to the ventricle. We don't know yet if they derive directly from radioglia cells, as they do express very similar markers to radioglia cells, or do they have some migrating intermediate astrocyte precursors that migrate up. And we also suspect that as other astrocytes are already present before them in the cortex, perhaps they can also derive to other waves of production of interlaminar astrocytes during development, because they keep increasing in density postnatally. So here they are, they arrive at the top and they start branching down processes that cross layer 1 after birth. So finally, we did a comparison of typical and rudimentary interlaminar astrocytes in development. So we took the mouse that have this very short rudimentary interlaminar astrocytes, layer 1 astrocyte in mouse. And you can already appreciate here in these pictures that from embryonic day 18 in mouse, so the gestation in mouse is about 19 days. They have these short processes, and the processes are growing up to P5, P12, but not much more after that. So they seem to reach a plateau of morphology much earlier than the primates. So this happens also for the density. So we couldn't find any appreciable difference in the density of these rudimentary interlaminar astrocytes after postnatal day 3. So the number really stayed kind of the same. And this is just to show you how instead in the primates, I was showing you that the density was keeping going up during development. And the morphology also, we couldn't find any difference after postnatal day 5. So the morphology, also the morphological complexity also reached a plateau much earlier. And we also compared the expression of the markers that we studied in the macaque. And we saw that they express only a subset of stem cell and differentiated astrocyte markers. So they do express PAX6, SOX2, and nestin, but they do not express CREAB, HOPX, and S1 and beta. So the take-home message from this is that the primate also express. The primate interlaminar astrocytes also have a typical expression of some proteins that are not expressed in the rudimentary interlaminar astrocytes in mouse. And finally, they do proliferate for a while too, but they do not proliferate after postnatal day 5. So to sum up this very last part, when we compare the typical interlaminar astrocytes and the rudimentary interlaminar astrocytes, we found different trend of density and morphological changes across development, and also only a partial overlap of markers, so both stem cell and astrocyte markers. So we concluded that mouse is not really a good model to study the interlaminar astrocytes. We should stick to primates if we really want to know why they're so special in primates. What do they do in primates? And in conclusion, so to conclude the second part of development, what I told you was that they increase their density, morphological complexity during postnatal development, but they are born prenatally, both in macaque and human, and they actively proliferate for a while when they reach their final destination, but after gestation day 123, they do not proliferate anymore, which is exactly the moment in which they start growing longer processes. And typical interlaminar astrocytes, they share only some features with the mouse layer 1 astrocytes. So we cannot really use mouse as a model for these interlaminar astrocytes. And these are, if you're interested in knowing more, these are the papers that we published along in the past 3 years on these astrocytes. So the first one is on the evolution on these astrocytes, and the second one, which was just published a few months ago, is on the development of these interlaminar astrocytes.